Assalamualaikum and good evening everyone. Today, we would like to present our topic titled Bioinformatics Resources for the Analysis of Phytochemical Metabolic Pathways. This presentation is presented by HANIS YASMIN, IMAN SAFFIYYAH here, and NURNABILAH SYUHADA. To begin, plants are often described as green cell factories because inside every plant cell, many chemical reactions are continuously taking place. Through these reactions, plants are able to produce thousands of special metabolites known as phytochemicals, such as alkaloids, flavonoids, and also terpenoids like we used to learn before, right? In fact, plants are estimated to produce more than 200,000 phytochemicals, many of which play important roles in medicine, agriculture, and also plant defense. However, studying how these phytochemicals are produced is not easy. This is because one phytochemical pathway is usually controlled by many genes, not just one. These genes can belong to large gene family and they may only be active in certain tissues like roots or leaves or under stress conditions. This is where bioinformatics become very useful. We can think of bioinformatics as a lens that helps us see what is happening inside the plant cell. By combining genomic data, transcriptomic data, and metabolomic data, bioinformatics allows us to visualize and predict these hidden metabolic networks. One of important bioinformatics resource is Phytosome. Phytosome is a plant comparative genomics portal that provides access to fully sequenced plant genomes. It helps researchers compare genes across different plant species and identify gene families and converse genes that may be involved in phytochemical biosynthesis. Another useful resource is the 1KP or 1000 Plants Initiative. This database contains transcriptome data from more than 1000 plant species. Using 1KP, scientists can study when and how phytochemical pathways evolve around different plant lineage. Finally, bioinformatics use co-expression analysis. The idea is so simple. Genes involved in the same metabolic pathway are usually switched on together. Databases such as ATAC2 analyze these expression patterns and help researchers identify candidate genes involved in phytochemical pathway.
Next, I'll be discussing some key bioinformatics databases that are commonly used to study phytochemical metabolic pathways. First, as you can see here, is an example from cake, showing the isoflavonoid biosynthesis pathway. So cake helps us see how basic metabolic pathways can lead to specialized compounds like flavonoids and isoflavonoids, which are important for plant defense and plant colors. Each box in the pathway shows an enzyme reaction, so it helps us understand how the metabolism flows. However, one limitation of K is that it is not specific to plants. So some pathways are predictions only and not fully confirmed by experiments. But it's okay, we have another database like Metasite to provide pathways that are based on real experimental studies, especially for specialized metabolism. So the information can be very detailed, which may be a bit hard for beginners like me to follow. And the other example is the Plant Metabolic Network, or PMN, which focus on metabolic pathways in specific plant species. Usually, researchers select a species first, then look at the pathways and the enzyme involved. And here, I chose Arabidopsis thaliana as the example. And PMN can also help find missing or unknown enzymes, or also known as orphan enzymes. Okay, next, for metabolite identification, GMPS is used to group phytochemicals based on how similar they are using mass spectrometry data. This helps find related or new compounds, but it doesn't show information about the genes. And the other example we have here is from KB, showing curcumin, which is a phytochemical from Curcuma longa, which is turmeric. So KB provides clear chemical names, structures, and so on, which is the basic properties of the compounds that we want. But however, it does not show biosynthetic pathways, so it is usually used together with databases like cake or Metasite. So finally, once we know the compound and the target protein, tools like AutoDocVina can predict how the compound binds to the protein. This links the pathway to its biological function. We won't show the docking life here, but it's an important step in research. So overall, these bioinformatics tools work together to connect metabolic pathways, compound identification, and how the compounds actually function in plants.
Hi, IMAN SAFFIYYAH here. For this part, I will show you the KEGG simulation on pathway mapping. First, we search for the KEGG homepage. I start the workflow by exploring the flavonoid biosynthesis pathway in KEGG. KEGG provides a map linking enzymes, reactions, and metabolites involved in a pathway. We select for MAP00941 because this is the reference pathway. This is a master template of all non-flavonoid reactions for all organisms. The power of KEGG lies in its organism-specific mapping. By selecting Change Pathway Type, you can get the organism-specific mapping on Arabidopsis genome to the global map. As you can see, the boxes turning green, which means it represents enzymes that is confirmed to be present in plant's DNA. You can select the green box and it shows you the detail of the enzyme. Pathway reconstruction. Plant site under PMN is used to generate pathway reconstruction. First, we look for the database. In this case, ARA site, which is the specific component, is also used. It stands for Arabidopsis thaliana. ARA site is more precise than KEGG because it includes enzyme isoform, multiple versions of an enzyme that may perform the same reaction but in different parts of plant like roots or leaves. Here, we can see the cellular overview of Arabidopsis thaliana. This is the structure of the metabolites. For this situation, we also can highlight the selected pathway that we want. For example, we want to see the flavonoid biosynthesis. You can see the red color appear, shows the exact pathway of flavonoid biosynthesis pathway. This diagram summarizes the bioinformatics workflow for analyzing flavonoid biosynthesis in Arabidopsis thaliana. The process starts from identifying plant sources, moving on to the genomic or transcriptomic data, followed by gene annotation to identify flavonoid-related enzymes. These enzymes are then mapped into metabolic pathway using KEGG, while plant site allowing pathway reconstruction and visualization. Finally, biological interpretation helps identify key enzymes and regulatory steps involved in flavonoid production. In conclusion, bioinformatics resources play a crucial role in the analysis of phytochemical metabolic pathways by integrating genetic, enzymatic, and metabolic data. Tools such as KEGG and PlantSight enable pathway mapping and reconstruction, improving our understanding of complex plant secondary metabolism. These approaches not only support fundamental phytochemistry research, but also offer practical applications in metabolic engineering and phytochemical-based product development. That's all from us. Thank you.